challenges for us to understand whether the technology per se is a problem, or it's the interpretation of the technology that itself is high fidelity. And that is why, we've got a debate coming later on today, for which I hope you will all be here, because that's an extremely important part, like everything else that we're doing. And all the challenges is prenatal diagnosis and recurrent pregnancy loss. So I want to really try to emphasize that it's your meeting. This is a meeting for you to challenge the speakers, to challenge what you hear, to create the opportunity and time to debate whether we believe we're going along the right path, or we believe we're doing the right thing. I think it was Ted Williams that said something that it's the only endeavor in humanity where in baseball, if you get three out of 10, you're doing really well. Well, actually, of course, we work in a field where if you're getting 3 out of 10, people are doing really well. We know the national averages in many countries, if you're doing well in the creation of a child from one of our IVF cycles, for example, is well below 3 out of 10. So whether we agree or disagree with people who are trying to find another channel to improve our live birth rates or not, what we have to all agree on is we need to do more, and we need to do it better, and we need to be more efficient. So I'm going to finish off just with a little quote from one of the great thinkers of all time, Albert Einstein. He said, it's not the fruits of scientific research that elevate us and enrich our nature, but the urge to understand, the intellectual work which we do, creative or receptive. And if that could be at the back of your minds today when you hear people get up at this lectern and offer their knowledge, enjoy their knowledge and challenge their knowledge and understanding, and let's try to move forward. And I'm not going to say any more. Antonio can't be with us today, so I've had to waffle on a little bit longer than I might like, but certainly that you would have liked. But it is, as I say, a great pleasure. And we started this movement about this meeting, this group, about 3 years ago, and I think it is very specific, and it's very important, I think, for all of us to try to challenge this field, because there's so much to do for ourselves, for our knowledge, but also for our patients. And so let's move ahead now. Let's create a little bit of space, bit of time, so that we can hopefully have longer discussions as we go on. And it's a really great privilege and pleasure to introduce my co-chair, but colleague and friend, Professor Yuval Yaron. Hardly needs any introduction, but I will just say very briefly that he's the Director of Prenatal Diagnosis Unit, the Genetics Institute, and Director, Pre-Implantation Genetics Diagnosis Laboratory at the Tel Aviv Sourasky Medical Center. And Yuval is going to talk to us this morning, just giving us a real basic background to introduce the whole of the 2 days, and it's the principles of genetics for non-geneticists. Yuval, thank you very much.
Welcome, everyone, to those who came back for the second and third time, and to new arrivals, and I hope you will enjoy this meeting. And I will start off by going over very basic principles. So for some of you, this may be kind of boring, or you already know this, but if even a few will learn something new, that will be helpful. And I will try to relate some of the slides to what talks we later have. So basically, genetics is the stuff of life. And in almost each one of our cells, we have the entire genome. One way of, so just before I start, these are my disclosures. So one way of looking at the genome is thinking of it as an encyclopedia with 23 pairs of volumes, where each chromosome is a volume, and each volume contains many chapters. Each chapter is a gene, which is basically a recipe for making a product needed for the body to maintain itself, to correct itself, and to reproduce. And just like any recipe is written in words and letters, so is the gene. The letters of the gene are A, C, T, and G. They're called base pairs because they are paired C to G and A to T. And there are about 3 billion base pairs in the genome. So each cell contains that amount of genetic information. That's quite a lot. The exome is only a part of the genome, and we'll hear from Mark Kilby tomorrow some talk about the use of exome sequencing in prenatal diagnosis. The exome is only about 1 to 2% of the genome, yet it contains most of the mutations causing human disease. And I don't know if you could see that, but the exome is actually only the part of the gene that encodes the protein. So it is only about 1 to 2%, roughly about 50 million base pairs. So that's smaller, but it enables us to look more in a more focused way, looking for disease mutations. This is basically what happens when the genome is transcribed, then translated to protein. Basically, the gene is transcribed into messenger RNA. The messenger RNA moves to the cytoplasm, and by the ribosome is translated into protein, and every 3 letters of the genetic code encode for 1 amino acid in the protein chain. The proteins are basically the building blocks of the body. So while the body contains carbohydrates and fats and so on, basically the proteins are either structural, they could be hormones, enzymes, and these enzymes metabolize different products and then create the other products that are in the body, like fats, carbohydrates, and so on. So when we think of genetic aberrations, basically something goes wrong with the code. It could be in several different levels. It could be at the level of the chromosomes. So that's like adding an extra volume to our encyclopedia or taking one out. So that's relatively easy to find. It could be submicroscopic. That's basically like tearing out a few pages from our book, from our volume. Or it could be a sequence variant that's like a spelling mistake in one of the letters. So chromosomal problems like aneuploidy could be detected by methods, cytogenetic methods, and we'll hear more about that from Vincenzo Cirigliano after me. So numeric abnormalities, basically like trisomies or monosomies, are some of the major causes of human disease, like Down syndrome, Edwards syndrome, sex chromosome anomalies, and so on. Mosaic aneuploidy is something that we'll hear more about from Santiago Munet and from Ariel Weissman and from Francesca Gratis. Basically, the fact that not all human cells are the same. Mosaic means that some cells contain different chromosomal structure. Could be cell lines that have trisomy alongside cells that are normal. It could be even more complex mosaicism. Mosaicism can arise by several methods, several pathways. For instance, they could arise from mitotic or post-zygotic non-disjunction, where you'd have normal gametes and a normal zygote, but then after cell division, some of the cells are normal, but then one cell is aberrant, resulting in Nondisjunction. That's just one chromosome instead of 2 in a pair, and trisomy. Usually the Nondisjunct cells disappear. So that's one way of mitosis, of mosaicism occurring. The other is meiotic non-disjunction with trisomic rescue. You have an abnormal gamete with 2 chromosomes. Can you see this? And after fertilization, you have a trisomic zygote, which continues into the embryo, but then sometimes one of the chromosome is lost, creating another type of mosaicism. And we'll hear more about that later. And mosaicism can create a continuum, beginning early in development, from the cleavage state onto the blastocyst stage, and then later on in the fetal placental unit. So this is actually a continuum, and a lot of discussion, we'll hear about that later on. The consequences of mosaicism depend on the chromosome involved. So some chromosomes are more harmful than other. It depends on the percentage of abnormal cells in the different tissue. It determines, is also determined by the tissue distribution, whether it be placental, fetal, or a combination of both. And there are other ramifications of mosaicism like uniparental disomy. I'll discuss that later. And also, using standard cytogenetic techniques, we can detect some structural abnormalities like balanced translocations, unbalanced translocations, and so on. When we dive deeper into the genome, we look for smaller changes. We can detect some submicroscopic changes that cannot be detected by standard cytogenetics, but rather by chromosomal microarrays, such as deletions, duplications. These are some of the known microdeletion syndromes like the George-Williams syndrome, Prader-Willi, and there's a really long list of conditions, and continuously more are being discovered. Such as, this is an example of a pathogenic copy number variant resulting in DiGeorge syndrome. But while we can now detect more of these conditions, we also detect variants of unclear significance, and that sometimes is a bit of a problem when one deals with in prenatal diagnosis, as well as in pre-implantation genetic diagnosis using techniques that can detect such small variants now. When we look at even deeper, we find sequence variants. That means that in the genetic code, one of the letters has been changed or has been deleted. And this requires sequencing technology that can detect such minute changes. In the past, we used to refer to these changes as normal and mutated. But things are not as simple. And the American College of Medical Genetics has created this new guideline for interpretation of sequence variants. And they range from pathogenic variants, what we used to call mutation, where you are certain that this is a disease-causing variant like a delta F508 mutation for cystic fibrosis. There's no doubt about that. To likely pathogenic variants, greater than 90%. But then most variants that we detect could be variants of unclear significance. We don't know exactly what they are. And this becomes really important when one begins to diagnose and try to predict the implications of such variants in human disease. And so are likely benign and benign variants. So what is a likely benign variant is just like having one letter change to another, but the meaning doesn't change. So you can write gray this or that way, it would still mean the same. So this is what a likely benign variant is. What a likely pathogenic is, one letter changes to another, but if this was a recipe for something, it wouldn't quite taste the same. So the implication of a pathogenic variant means that something has changed in the sequence, therefore something has changed in the protein. And this is just one example of the protein for cystic fibrosis, the CFTR, which is basically a protein channel that allows chloride ions to leave the cell into the lumen of, say, the respiratory tract. And if the mutation changes the protein just by a little bit, This stops the chloride ions from leaving the cell, and therefore not allowing water to leave the cell into the lumen, creating viscous mucus that creates the disease. So it is the even small, minute changes the meaning of the protein, and therefore causing the disease manifestations. When we look at prenatal diagnosis of human anomalies, in the past we had very rough tools, so we could use karyotype or rapid detection methods like QFPCR. This allowed us to detect a greater percentage than in the past. So at least 30% of fetuses with malformation can be detected using standard cytogenetics or rapid modes like QFPCR and karyotype. Copy number variants add about 3 to 6% over a standard karyotype. But if you use that as a first line, it can detect most of these abnormalities. Now, but it turns out that a great number of fetuses with abnormalities cannot be detected by these techniques because their condition is caused by sequence variants. And to be able to diagnose these, you would need more robust techniques like whole exome sequencing or now whole genome sequencing. And that, depending on the choice of patients, you can detect an additional 6 to 80%, or let's say roughly 30%, additional abnormalities in fetuses that could not have been detected up till now. So, as I said, we'll hear more about that tomorrow. Just a few words about modes of inheritance. Autosomal dominant inheritance is caused by a mutation or a pathogenic variant occurring in one parent. In one parent, where that parent has one normal copy and one abnormal copy, and the chance of transferring that mutation to the offspring is 50% irrespective of gender. And this is a typical pedigree of an autosomal dominant condition. Here are some examples like neurofibromatosis, tuberous sclerosis, achondroplasia, and so on. Autosomal dominant conditions are inherited, but sometimes they may occur de novo. So, for instance, 80% of achondroplastic are a result of de novo mutations. Autosomal recessive conditions, in autosomal recessive condition, each parent of an affected child has one normal copy and one abnormal copy, and there are 4 possibilities: having an affected child that inherits both abnormal copies, 2 out of 4 are carriers carrying either the paternal mutation or the maternal mutation, and 1 in 4 is unaffected. And this is a typical pedigree of an autosomal recessive condition. A lot of times you would see that the parents are related. This is more common in consanguineous mating. We'll hear a lot more about this from Jim Goldberg tomorrow when he talks about carrier screening for most of these autosomal recessive conditions. And here are some examples like spinal muscular atrophy, cystic fibrosis, Tay-Sachs disease, and the list goes on and on. Now we have the ability to screen not for 1 or 2, we have the ability to screen for hundreds of different diseases, and screening for these conditions allows us to prevent the birth of affected children either by early prenatal diagnosis or by PGD, and we'll hear this later on by Rebecca Zimmerman. X-linked recessive conditions are caused by mutations in genes on chromosome X, where the carrier mother has one normal and one abnormal copy. The normal copy usually compensates for the abnormal copy, therefore carrier mothers are usually healthy. And half of the boys receive the normal maternal copy and will be healthy. Half of the girls will receive the normal copy and will not be carriers. but half of the girls will receive the abnormal copy, be carriers, and half of the boys will receive an abnormal copy and will be affected. This is an example of a pedigree for an X-linked recessive condition, where you see only affected males and they're all connected to each other through maternal lineage. And here are some famous examples like Duchenne or Becker muscular dystrophy, hemophilia A and B, and so on. So one point to remember with X-linked recessive inheritance is the fact that carrier females may sometimes be affected, albeit to a lesser extent, because of non-random X inactivation. And lastly, X-linked dominant, where irrespective of the gender, any person carrying the mutation, be it a male or a female, will be affected. And here are some examples like X-linked hypophosphatemic rickets or Alport syndrome. There are other forms of inheritance which are called non-Mendelian because they don't follow any one of these modes of inheritance. One of them is multifactorial inheritance. And basically, a lot of human conditions are caused by multifactorial inheritance. This could be a combination of different alterations in different genes or alterations in different genes in combination with environmental factors. Some known examples are pyloric stenosis, neural tube defects, and so on. But basically, if you think of any human conditions like cancer, neurodevelopmental problems, neurodegenerative problems, and so on, can be caused by a variety of different conditions. And in this model, we think of a threshold where you need a certain number of factors that will combine, and once the threshold is passed, you would have the disease manifest. It's kind of difficult to predict what are the chances of having an affected child, but because some of these factors are still unknown, but we're trying to recognize more of these. Another form of a non-Mendelian inheritance are triplet repeat expansions. This is not a mutation per se, it's not a change. These are caused by triplets, triplet repeat like CTG repeats, ACG repeats, CGG repeats, and so on, whereas normal individuals have a small number of repeats, and as the number of repeats expands or grows during cell division or during meiosis, then a certain threshold is passed and disease manifests. One example is Fragile X syndrome, where normal individuals have 50 to 55 CGG repeats in the 5' untranslated region of the FMR1 gene. Pre-mutation carriers have up to 200 repeats, and individuals having more than 200 repeats will have the Fragile X syndrome. So pre-mutation carrier females are at risk of having children with an expansion of the number of repeats, so they are at risk. Other examples are myotonic dystrophy or Huntington's disease. Another concept that is being recognized is the concept of genomic imprinting. We used to think that the two copies of the gene, the one inherited from the father, the one inherited from the mother, function in the same way and are expressed in the same way. But this is true for most, but not for all genes. Some genes are imprinted in such a way that they are expressed only from the maternal or the paternal copy. Imprinting implies that one copy is silenced, usually by methylation. And imprinting has to be erased so that it is reinstated during meiosis. So in the germline, the parental imprint is erased and then reestablished. But once it's reestablished, it is maintained through mitosis until the next generation. And one implication of imprinting genes are uniparental disomy. This means that a certain chromosome is not inherited from each parent as it should be, but rather both copies of a particular chromosome are inherited from the same parent. And this may have implication if this chromosome has imprinted genes on it. And I'll show you just one example. This is uniparental disomy caused by trisomic rescue. So just imagine you have a disomy 15 oocyte, where you should have one copy of chromosome 15, but if you have two copies of chromosome 15 in the oocyte, and this oocyte is fertilized by a haploid sperm, you get a trisomic 15 conceptus, having two maternal and one paternal copy. This would result in a miscarriage usually, but if there is a trisomic rescue, one of the chromosomes is lost, then this conceptus can continue to develop. In 2/3 of the time, the extra maternal chromosome is lost. Then you have a state where you reconstitute a disomic conceptus having one maternal and one paternal copy. This is okay. But in 1/3 of the time, you lose the paternal copy and you remain with two maternal copies. Since there are imprinted genes on chromosome 15, this would result in the Prader-Willi phenotype. Here, the two copies of chromosome 15 are not identical. They're heterodisomic. They're different, but they're both maternal. Another form of uniparental disomy may result from a nolisomic 15 oocyte. This is an oocyte that lacks chromosome 15. And when this would be fertilized by a haploid sperm, you'll get a nolisomic conceptus. Again, this would either not develop or result in a miscarriage. However, sometimes this one chromosome is duplicated in the cell, resulting in a correction of a monosomy, and this leads to isodisomic uniparental disomy of chromosome 15, meaning that two copies of chromosome 15 are paternal and are identical, and this will result in Angelman syndrome. So we see this is a different mode of inheritance, not usually. And the last mode of non-Mendelian inheritance is mitochondrial inheritance. One thinks of the mitochondria as the batteries of the cell that create the energy, and each one of those mitochondrion has several DNA molecules. They're circular. They contain several genes. They're about 16,000 base pairs long. They encode for polypeptides that are associated with energy production using oxidative phosphorylation, and they are inherited exclusively from the mother. So the few mitochondria that are in the sperm get lost after fertilization. So when there are perturbations in the mitochondrial DNA, this may lead to diseases affecting the nerve, muscle, and so on, and they are usually transferred only by the mother. Some examples include mitochondrial myopathy, diabetes, and deafness, and so on. So I think to sum up, this is just a brief introduction into the topics that we'll be discussing. And if you have any questions, this is the time to ask. So you have now graduated Genetics 101, and we're ready to proceed with the next talks.